Okay. Hello everyone. Today we'll be presenting our primer design project for BSN 3202 Genetic Engineering. My name is Nur Akila binti Adlisha and my matrix number is 229748.
My name is Yasmin Shazwani binti Muhammad Safwan and my matric number is 231055.
Hello everyone, good evening. My name is Lim Roshan and my matrix number is 230210, and I will first discuss the primer that our group designed. So the gene sequence of the bacteria that we are looking for is from the Escherichia coli, structure K12, substructure MG1655. And we have designed 3 pairs of primer to amplify a certain gene, and the gene that we are going to amplify are beta-galactosidase permease, thiogalactoside transacetylase, and chromosomal replication initiator protein DNA. So first, I will start with my primer, which is the primer design used to amplify this beta-galactosidase permease, which is a leg Y in Escherichia coli. So first, this sequence is the sequence of leg Y gene. But I purposely make the sequence ranging from the base pair from 361880 to 36220, where the highlighted region over here is the gene that will code for the leg Y gene. So first, the primer that I designed the forward primer that I have designed is this one 5 prime CGCAT CCGA CATT TGC, and this primer can be found in my sequence over here, which is from here, this CGC onward, and until this before I highlighted from before the highlighted T here. So it is a length of 19. The primer length is 19 base pair, and the TM value is 57.85, which is still in the region of 55 until 58. This TM primer does not exceed 60 degrees Celsius to prevent the non-specific annealing of the primer. And then the GC content of my forward primer is 52.63%, which is still in the range of 45 to 60. And this GC content is important to ensure the stability of the primer to the template. So from the forward primer and the 5' primer over here, we can see the 5' has a higher GC content than the 3' end. So this this can prevent the this can prevent the primer from not form a primer dimer formation. It can enable this primer to bind specifically to the template. And this self-complementary, meaning that the region in the DNA itself, the template DNA, that can bind to itself to form a secondary structure and a hairpin structure. So the value of my primer in self-complementary is 4. It is still low, but it is recommended to have a lower self-complementary over here. So the self-3' complementary is 2, and this meaning the ability of the primer of my primer design to form a primer dimer with another primer. So the value is 2 is still low, and it is still acceptable. So next, it is my reverse primer. Reverse primer, as I design, is 19 base pair long, and it can be found in my sequence over here. Let me show you. Yeah. It is from this T. Well, but we need to use the reverse complementary to find this sequence. So it is from this T to this G over here. ACG. Yeah, it is from the T over there to the G over here. So this primer, the TM value is 58.31, which is almost close to my forward primer, and the GC content is the same, but in the sense of self-complementary, it is lower than the forward primer, meaning that it has low ability to form a secondary structure or hairpin structure, which is better for a primer. So this is my calculation on the TM value, and the TA value is the annealing temperature, which can be calculated by subtracting the TM value with 5 degrees Celsius. And this figure shows how my primer will run, and at the end of the day, my PCL product will be leg Y, and then the length is 1307 base pair, which is a complete leg Y jean. So that is from my part.
Next, I will continue for the second sequence, which is leg A jean. So firstly, I want to explain the function of leg A first. So leg A jean also include as thiogalactoside, transacetylase. Okay, just like Lucian said before. This gene help to protect the cell by detoxifying non-metabolized B-galactoside, and it does this by transferring an acetyl group from acetyl-CoA to these lactose-like molecules, which may aid in their removal from the cell. Unlike LAC-Z and LAC-Y, the LAC-A gene is not required for lactose metabolism, and bacteria lacking LAC-A can still utilize lactose normally, suggesting that its role is supportive and protective rather than essential for energy production. Okay, can you go down? Bawah lagi. Okay, so based on this table, we can see that this is the forward primer that is used, which is 20 base long, and it is bind in the correct region of the gene. Also, the melting temperature here is 59.90 Celsius. So an annealing temperature of 54.9 was chosen to allow proper binding. And the reverse, can you go down? Okay. And the reverse primer in this table that we choose is also 20 bases long and has a melting temperature close to the forward primer, which is 60.39 Celsius, which help both primers work together efficiently during PCR. And for the GC content, both primer contain 55% and low self-complementary, meaning they are stable and are likely to form unwanted structures such as hairpin or primer dimers. Bawah lagi. Okay, atas. The calculation. Okay, so for this one is the calculation on the melting temperature. So as you can see from the table, and the calculation is a bit different because the TM value in the table, we use the computer software that applies more accurate methods, taking into account factors such as salt concentration, magnesium ion, primer length, and best pair interaction. While the manual formula that I use here is okay, in this slide. Okay, it's only a simple estimate, does not include this variable and give a slightly higher value. Okay. Okay, you pergi dekat sequence yang atas. Okay, so this is the sequence of leg A that we choose. Okay, so that's all from me.
Okay, so for our final gene, I will be presenting the primer design. So I choose DNAA in Escherichia coli. The reason why I choose it because the gene is a critical target as it encodes the initiator protein for chromosome replication. And after evaluating several options, I have selected primer pair 9 as the best performing candidate for the following technical reason. So Scroll bawah. So dekat table. Okay. So the primer sequence for the forward primer is TAT, TCA, AGA, AAGA, AACG, GTG, GCGG, and the reverse is CTC, CGG, CAG, ACT, GTG, GTT, AG. So basically, both of these primers are exactly 20 nucleotides in length, which is the standard for maintaining high specificity during qPCR. And for the melting temperatures, it is exceptionally well balanced with the forward at 59.83 Celsius and the reverse at 60.11 Celsius. And to satisfy for our requirement for TA, we calculated the aniline temperature using the formula Tm-5, which gives us the TA of approximately 55.1 Celsius. So it has ensured efficiency and synchronous binding for both of the primers. For the GC content, it is 55% for the forward and 60% for the reverse, which falls perfectly within the optimal 40 to 60 range. It also provides stable hybridization without making the strand resistant to denaturation. So in terms of secondary structure, why the major factor in choosing pair 9 because of the stability of the ends. Because the self 3' complementary is only 1 for both primers, this is extremely low value because it's vital because it prevents primer dimer formation at the 3' ends, which would otherwise interfere with our qPCR quantification. And for the applicant size, the predicted PCR product is 139 base pair. In qPCR, the applicants between 70 and 150 base pair are preferred because they amplify with higher efficiency and yield more consistent result. So that's all for me.
So for the LAC-Y gene, actually it is responsible for the transfer of lactose. The important transport the lactose to the cell to be metabolized by beta-galactosidase, which is also an essential gene in the Escherichia coli. So I think that's all from our group. Thank you.
Thank you.